I accidentally sent out an email to everybody. Um, okay, I think I'll start now. So does anyone have any questions in particular? So I'll kind of like leave it open right now for any questions. Either I can answer or any of your classmates can answer. But keep in mind, if you do answer someone else's question, make sure you're like sure about the answer so that no one's kind of like, on the fence with anything. But if anything, it's on your book, it's in lecture and things like that. Okay, so I'm not going to be going over what you went over last class because that's not why you guys are here. So let me just kind of start with some reminders. Your test is in 3 days. It's on Monday. It's from 5. It should be 9 p.m. So it's going to be open from 5 to 9 p.m. Like for people who like have to work and stuff, she kind of adjusted that. Okay, SI exam going right now. We're hiring if you want to do what I do, blah, blah, blah. So normally in my normal sessions, at the end of them, I provide these mini quizzes that I get questions from myself, I make from lecture or based off your book, like from your book, word for word. I have those up in a section for under session materials. So they don't have answers right now, but you can always use that to test yourself, just extra material. I will put answers to those later tonight, hopefully. So look out for that. And then here I put don't forget random figures. I think you guys have realized by now that your last exam, she might put something random from like a figure that you may have overlooked. So just as a reminder, kind of go through those figures so that you kind of understand these different organisms or just these things that you wouldn't have expected before. Because again, anything is fair game. Okay, so now I'll just kind of start. I'm going to start off where I left off on Wednesday. I try to break it half and half. So I'll start off from there. I'm pretty sure I'll have time since I didn't go over anything from last class. So I'll start up back at the beginning again for those who may have missed it. So let me find it. Left off on sensory. I actually might start at ear just because I think it's pretty. Actually, we'll just start at sensory. Why not? Okay. So I'll start here and then we'll kind of get through everything I have. A total, I have like 125 slides. Obviously, I can't cover everything that you have, so just keep that in mind. Your TAs have sessions over the weekend. Just use this as like if something you don't recognize, you can use this as a reminder to go back in the book and kind of brush up on it if you don't recognize it. So use this as a checklist if anything. Okay. So yeah, thank you for whoever's doing this. I usually try to leave some blanks in there so that people can kind of interact. But these are kind of your main receptors. You have 2 other receptors as well. Actually, can you guys hear me? Can anyone reply saying you can hear me just to test this out?
I can hear you.
Okay, cool. Thank you. So yeah, these are some of your receptors. You have 2 other ones also, your electroreceptors and your magnetoreceptors. So kind of understand that as well, but that's on a different slide that I made, but these are kind of your main ones. Try to associate with an example if you can. It makes things a lot easier. And just kind of how they work. So if we go through them right now, your mechanoreceptors, anything that deals with pressure and physical forces. So for example, things in your ear. That's a really good example of mechanoreceptors. I put echolocation. That's kind of like a random thing that she might throw in because that's what bats have. So again, fair game for anything like that. And it can be involved with things such as touch and hearing. So out of your 5 senses, that's kind of what everything here is encompassing. Photoreceptors, just light. We'll go over parts of the eye later as well. Chemoreceptors. Photoreceptors also realize how like certain things are active in the light and some things are active during in the dark. Chemoreceptor. You're free to like screenshot any of this as well. Chemoreceptors are associated with your smell and taste, so you have different ligands and molecules that can bind. Thermoreceptors are just change in temperature, so like things on your skin especially, you can detect changes in temperature. Nociceptors are for tissue damage, so just any damage that you have in your skin or internal organs. Proprioceptors, I would just mostly say maintaining your body balance and equilibrium. So it's like you're able to coordinate the whole like rubbing your head, patting your stomach kind of deal, or like touching a tip of your nose with your eyes closed. You're able to do all that because of like you're being aware of where things are in your body. So that's kind of dealing with your proprioceptors. And if anyone has any questions, just throw it out whenever or put it in the chat. And I'm just going to talk. Okay. These are some of your mechanosensors. So I'm looking for kind of specific ones here, but you have these receptive fields on your body. So your fingertips have small receptive fields and then your back has larger ones. So a way to test it out, there's this toothpick test. I don't know what it's actually called, but you take 2 toothpicks, you put them to where they're touching in the middle at one point, and then you slowly go further and further. whenever you feel that there's two separate toothpicks, that's kind of how you can detect that change. So on your fingers, you can probably detect it with 2 millimeters. On your back, you can probably detect it with like 2 centimeters or 2 inches apart, where you can detect a difference. So that's a good way to test your receptive fields. And then here, I'm also, yes. So for this, you have 4 mechanosensors that was listed in your book. You have Meissner's corpuscles, Ruffini endings, Pacinian corpuscles, and free nerve endings. They each have different response, or what they each respond to different things. So your free nerve endings are for light touch. Your Meissner's is light touch also, but also vibrations. And then these two are deep pressure. I have an image on the next slide that'll make more sense. So let me just show that. Okay, so this is kind of an image, and if you look at this, you'll understand why they are the way they are. So up here, these are your free nerve endings, and then these are your Meissner's corpuscle. That's why they detect light touch. They're at the surface of your skin. Whereas Pacinian corpuscles here, and then your Ruffini endings down here, it's a little lower, it's deeper into your skin and your dermis. So that's kind of why those detect deeper touch, because you kind of have to push in more, and then you have these that are more light touch. That kind of makes sense. I put another image here, right here, your free nerve endings. It just kind of says it can detect pain and temperature. So that's kind of a way of it detecting, like using your thermoceptors or even your nociceptors. So you can kind of think of it like that as well. Obviously, it's not too specific, just kind of where they are, what they respond to is, I feel like, is pretty good enough. And then at the end, if I have time, if you want me to go back to a certain slide, let me know, just kind of mark it down, because I do have a lot of slides. Okay, so your vestibular apparatus is very important, especially in terms of when you're moving your head. You have fluid inside that can detect, that will like brush over these hair cells and things like that. So let me just kind of run through it. So this is like a general vestibular apparatus, just saying what it is. The function of it is to detect rotational movement and just positioning of your head. So every time you move, you have fluid that's moving inside your ear. The components of it are your semicircular canals, utricle, saccule, and endolymph. So your semicircular, you have 3 semicircular canals. So if you look at this image here, you have an anterior, lateral, and posterior, and those kind of cover all your planes, your X, Y, Z planes. So that's kind of a good way to detect your spatial awareness. And then within that, you have ampulla and stereocilia. Also, we'll go into the depth of that later, so don't get confused. I mean, you guys should know this by now. Utricle and saccule, your book doesn't say exactly what they do, but I just kind of looked it up because I like to know where everything is and what they do. So utricle is for horizontal acceleration, saccule is for vertical. And then endolymph is the fluid that's actually moving around in response to your own movement that you're doing yourself. So going like this and things like that, endolymph is currently moving. Yeah, okay. I feel like I'm talking fast, but just let me know. And then this is just an image. So your vestibular apparatus is this whole thing. This is also part of your inner ear, so we'll get into that later. So if we focus on just your semicircular canal, if you look over here, this is like a blown up picture of it. It's going to blow up to this ampulla, and then within your ampulla, you have all these different components, right? So this is your cupula, this gel-like cap. Inside your cupula, you have hair cells, and those hair cells synapse and will go to your afferent neurons to go to your brain. And then, so every time you move, this endolymph, that where this arrow is moving through your head. It's going to brush past this cupula, and then it'll cause the hair inside that cupula to move somehow. It'll move, and then you'll have these ion channels that open. I have a picture of this later that's really good, so I'll just show that later, but keep this in mind. So as these hair cells open and move, it'll cause depolarization, and that signal will be transmitted down into these neurons. So you have afferent and efferent. Definitely know the difference, because that kind of is important in terms of what it's going to do. So afferent, I've said this a lot, but I'll just kind of say it again. So afferent, I think of it's going to go towards you. So every time you sense something from the outside, like your mechanoreceptors or whatever, it's going to send that signal, and it's going to go towards your brain. Efferent is when it's going to go out, away for you to create a response. So efferent, I think of exit. So that signal is going to exit away from your brain to tell your body or your muscles to do something. So it's kind of like an in and out. So in this case, since we're talking about senses, you're going to have some sort of stimuli that's going to hit a receptor, and that's going to go send that signal to your brain. Your brain's going to integrate the information and then send it out to tell your muscles or whatever to do something. So afferent and efferent. So keep in mind the neurons, because you're going to see that a lot with all these senses. Okay, so then that's for your semicircular canal. Then you have your utricle and your saccule. So your utricle and saccule have these things called otoliths. These are your calcium carbonate crystals. So every time something moves, it'll move those crystals, and those will also end up moving your hair cells. And then in those hair cells will cause depolarization. And as you can see here again, it's going to just go to your brain. So it's that whole system of just going towards your brain to integrate that. So associate somehow otoliths and utricle and saccule so that you just don't remember. So it's good to, like, if you see that, you'll recognize it, bam, you can make a connection. Yeah, so that's really good. So make sure, I have that later on. So statocysts, they have something called statoliths. I'll actually go into that into the next slide, so I don't want to confuse everyone, but I will go over that, and that's a really good connection to make. Okay, so now we're going to talk more about your semicircular canal and just specifically. When I talked about hair cells, this is what I'm showing. So this is a really good image in my opinion. So this is when you don't have any endolymph moving or any like really movement going on. So as you can see here, these are the channels that would open and close. They're closed right now because there's no movement to make them open. This is an image that shows that when you are moving, so your hair cells are going to move, and that'll open these channels by moving it this way. And then the potassium can go in, and in turn, calcium can go in also. And then you'll create a depolarization effect. So I like to make some kind of flow chart. So what I would say is like endolymph, stereocilia, potassium, calcium, depolarization. Just make a flow of connections like that, and it'll make it easier to like remember things. But that's just my way. There are a lot of different other methods. So I think this is really good to understand stereocilia, and this will be seen a lot in terms of sensory information. So you have your ampulla here. I kind of said the same thing. Endolymph is going to move, brush over your cupula, and basically you want to get your hair cells to move to cause that depolarization. Depolarization is with all these ions moving in and out because, again, action potential. You guys know how that works. And then I can go over that again, but that's going to be later. Yeah, so that's that. I think this is everything I said, but I said it, but this is in words. So I'm not really going to read that again unless you guys want to screenshot that or whatever. I put a lot of these pathways in there, but that's kind of how I think. So, okay, so now we're going to talk about utricle and saccule. So the function of them is to provide information about position of the head with respect to gravity up and down and for changes in linear movement. So this is kind of between, if you remember back to that image, it's between your semicircular canal and your cochlea kind of. And they also have hair cells of their own. So we talked about the otolithic membranes. That's the gelatinous covering. And then the ear stones or the otoliths are. Oops, sorry, whoever was drawing. This is more of the mechanics. So that was like the broad components. Now we're going to get into mechanics. So every time your head moves, the things. So this should be otolith, same thing as otoconia, whatever. I just got this from some video. It's basically going to move, and all those things will cause the stereocilia to bend and open. So it's basically the same idea there. This one's more with otoliths. Yeah. Okay. These are just more images that I found. So this also shows these tipplings or the stereocilia when they open, goes in, depolarization. Calcium also goes in because you need that. This is another image of the otoliths and all that. And yeah, so it's good to find different images because some may show differently, but they all have the same general function. Okay, so as someone mentioned earlier, so statocyst, and then we have lateral line system. So this is kind of how other organisms and animals are able to sense their surroundings or their own sensory system, really. So fish have something called lateral line system. It can help them detect prey, avoid predators, obstacles, currents, and things like that. It's just similar to our own vestibular apparatus, but it's kind of the fish's own way of detecting where things are. So out of all this, I would kind of say neuromass is kind of more specific to them. So if you ever see neuromass, then you're able to associate that with lateral line system. So it's kind of a quick recognition. They kind of have the same idea here. They have stereocilia, same thing. Example, school of fish like a Nemo. Statocyst, as someone said earlier, that was a really good connection. You want to be able to make those connections because it's a bioclass. So it's not just humans, it's animals as well. So they have something called statoliths, and it's similar to our otoliths. So that's kind of their way of detecting movement as well. Yeah, everything I already said. I have to double check myself so I don't miss anything. Yeah, so the lateral line, they have that one lateral line that goes through their body. So yeah. And this is just some images. Again, this is in your book, so I'll just go past that. Oops, sorry about that line. So now we're going to talk about parts of the ear. So down here, these are supposed to be arrows. So I kind of put in order from like outside to inside. So this is kind of one way you can think about it if you would like. But so the outside of here are the parts that you can see. So your pinna is like the actual flap that you can see. Auditory canal, if you stick your finger in, that's one way. Your tympanic membrane is another word for eardrum. Inside you have ossicles. Make sure you know the function of these ossicles, and then we'll go in. Well, I'll go into depth with that later. This is kind of a brief overview. And then you have another membrane separating middle from inner, and then that would be your oval window. And then you have your inner, and that's all your semicircular canals, and then, well, you know, all those things. You also have your cochlea, which we'll probably talk about more. These also have other names. I put that here. So just kind of recognize it just in case she throws it in there and you won't be thrown off guard. Yeah. Okay. Your outer ear is to concentrate and focus sound waves. So that makes sense because it's kind of giving it a direction to go through. So that's that one-way canal. The role of your ossicles are to amplify a signal. So it kind of makes it stronger for your cochlea to be able to perceive it. And your inner ear just has those hair cells for the action potential and all those things like that. Okay, I'll give it a couple seconds. I don't want to take y'all's. Okay. This is just another image. This also just shows kind of like where the order is and same thing. Okay, so now we're going to talk about invertebrates real quick. They have something, if I were to associate invertebrates, I kind of would just, if you see tympanum, that's kind of a good way. It's kind of a different word, so that's kind of how I would make that connection. So the way invertebrates do it, they're just a little, not as complex as ours. So they detect most of their ways through like anything on their skin or the surface. So they have something called a tympanum in there. There's an image in your book, so I would look at that. I just don't have it right now. And then cephalopods have something similar to the lateral line system in fish. So basically, all organisms have some sort of way to detect their movement and awareness in like where they are. So for invertebrates, tympanum I think is for some insect. I remember. There's a picture of that. Cephalopods are things that look like squids and octopus, something like that. I'm not like a fan of like, I can't memorize all these animals, but I try to recognize them. This is the same thing here. And then eustachian tube. I think this is something that I didn't cover, but I can talk about it. She just kind of said it allows, so if you get congestion, like if you're sick, it's kind of blocked. So it's harder for like, you can't like breathe or something like that. It's like different, especially when you go on airplanes or yawn or something like that. You open it to allow that tube to be open and you can perceive things better. I don't remember too many details about that one. So again, this is something that you should go back into. Don't forget something like this. This is like a small detail, although it's in the book. Yeah, I don't think I covered that, so make sure you do. I didn't put a description in here because I forgot, but this, I just told you guys what it is. It's your external parts. Allows you to concentrate and focus sound waves into like a direction, which would be into your ear. Okay, so from now on, it's going to go in order from outside to inside so that you guys kind of get a feel for that. Okay, so after you hit your outer ear, now you have this kind of, like a direct separation, but it's kind of telling you you're exiting outer, you're going to go into middle. So you have this tympanic membrane, also known as your eardrum, vibrates in response to sound waves from the outer ear. That's all I can really say about it. There's not really much to it. Next would be your middle ear. So you have ossicles, they function to amplify and enhance the signal. You have malleus for hammer, incus anvil, stapes stirrup. Your stapes is the last one at the end, and that one is connected to the oval window. So that kind of will tell you it's going to go into the next phase. And then when you hit the inner ear, everything is like fluids from there. Yeah, that's one way that you can do it like that. Yeah, any way that helps you memorize would be great. Oval window, not really anything else either. Okay, so you have your inner ear. This is like covering some of my things, so hopefully. Okay, so this is more focused on your cochlea, just like the hair cells inside your cochlea also. So your cochlea is made up of these three sections. If you look here, this was your vestibular canal, you have your cochlear duct, and then you have a tympanic canal, which is down here. But your fluid just kind of goes out, goes in and then out, and then you have a round window. But similar to the previous senses that we talked about, or with like the hair cells, really, as it brushes over, it's going to allow these hair cells that are embedded in here inside your organ of corti, as you can see here, to go out to your afferent nerve, and it can go to your auditory cortex or whatever cortex or part of your brain that it's supposed to. So say you'll get a signal from your ossicles, it's going to get amplified here so it can go stronger and send through your cochlea. Your basilar membrane is going to vibrate down here, and then it's going to cause the hair cells and your tectorial membrane to move. And that movement will open those stereocilia or those hair cells, allowing for depolarization and allowing for an action potential go out to your afferent and then go to your auditory lobe or auditory, whatever it is. Anything to your brain, wherever that signal is supposed to go. Okay, so this is basically your cochlea is supposed to be like unwounded. And I kind of did the same thing here, just made like a pathway. Again, those are supposed to be arrows. And you're free to make whatever notes you would like as well. I don't care. Okay, next. Again, this is another picture of your hair cells. This is something called basilar tuning, or if you take psychology, I think it's also called place theory. But basically, in your cochlea, you have different parts that can detect different frequencies. So that's kind of how you were able to sense different things. So how I like to think of it, so this one has higher frequency, and this one has a lower frequency in terms of what they can detect. So if you have a high frequency, I think of it as like it's a lot, so they probably have a higher amplitude. That's why I can go here, because it's more wide here. Whereas lower frequencies, they probably have longer wavelengths and smaller like amplitude, smaller this, so they're able to go in deeper. I'm not positive on that. I've heard it before, but I'm not sure if it's like, I feel like it might be right. But again, that's a good way to remember how it can travel inside. So lower frequencies will go in more. Lower frequencies have longer wavelengths, and they're kind of stubbier, I guess, shorter to where they can fit through the narrow space because it gets more narrow here. So this is known as basilar tuning, and this is how your ear can detect different frequencies. Okay.
The farther you go in cochlea, the higher the frequencies. I thought it would be farther you go into cochlea, the lower the frequency.
Yeah, the further you go in, the lower the frequency. Yeah. How did I word it? That was.
Oh no! Someone just wrote it down, like on the page.
Okay. Yeah, so it's more the lower you go in, the deeper you go in, it's lower frequency. How I like to think of it, just like the different sizes if you think of the waves. So like these waves are probably higher frequency, and you can think of like these waves where they're fatter and less frequency. The fatter ones are shorter in length, so that they can go in deeper. That's how I think about it. But it makes sense with this as well if it helps you. Did I just do this? I think I just did this. Or I added more down here, but it doesn't matter. Yeah, so afferent nerves. Know the thalamus. The thalamus is one of the ones that have that synapse with a lot of the sensory information. So we're going to be seeing that a lot. Okay, now we're going to get into vision. So these are kind of like the different types of eyes. So this is simple eyes or single eyes. Simple eye. This is simple eye, and these are kind of in organisms that are very, like, they're just like worms or something like that. Their bodies aren't as complex as ours, or even like a cephalopod. These are compound eyes, like flies, so they just have a bunch of these little eyes, and each of those eyes can detect a different image. So that's why every time you move, they're able to catch it so fast and evade and leave, because they can see that movement in their many eyes. And then you have this. This is a cephalopod. Their eye structure is very similar to our own, the vertebrate system. So you'll kind of see how they align. We have all those parts as well, if not more. And I have a description of everything I just said on words. So out of this, okay, so for simple eyes, they have something called an ocellus. So that's not a bad thing to remember as simple eyes. Compound eyes have something called ommatidia. What else is there? I also did a pathway for that. Single lens. So change the amount of light allowed to enter and focus the light with lens. That's the same thing with us. We have that same thing as well. Know what accommodation is? Just because, like, it's like a vocab word. It's like a big word. You should know what it means. So accommodation. So if you have things that are closer to your vision, your eyes have to stretch more inward and contract. But if things are further back, your eyes are more relaxed. But basically, the whole idea is that your eyes are able to adjust, like turning inward and outward whenever needed, depending on what you're focusing on. But again, you can look it up just for more details. I try to sum things up for today. Oops, sorry. So this is the image of your eye. It does tell you each of the functions. So I personally also like to know what each of these things are, like sclera, choroid, retina. So for time's sake, I'll just kind of show this. This is that same thing, but in words. So let me just go back to this so that you guys kind of visualize. So your ciliary body has something called ciliary muscles, which is here. Those ciliary muscles control the iris. The iris is the color part of your eye. Inside the middle of your eye, that black thing is your pupil. So your iris is the thing, with the help of your ciliary muscles, it'll allow the iris to contract or dilate so that certain amounts of light can enter your pupil. So for example, say you're outside on a sunny day and it's super bright, right? Your eyes are going to, your iris is going to contract. So it's going to get smaller so that not too much light is entering your pupils. Whereas in the dark, it's going to dilate because there's not even that much light around you anyway. So that's kind of how your eyes will adjust with your ciliary body and your iris. Your cornea is this front part, and then the back part, like the end of the cornea, is like your sclera, like covers 5/6 of it, and then your cornea covers like 1/6 of it. You have aqueous humor and vitreous humor. So aqueous humor is more fluid, and it kind of covers from this portion. So if you can see this here, so it goes from your cornea to your lens, and then your vitreous humor. Your vitreous humor is like a jelly-like substance. It goes from your lens to the rest of your eye, and that provides for nourishment and support, which makes sense because it's trying to like keep it, round and all that. Your optic nerve, aka, kind of connects with your blind spot as well. That's where you don't have any photoreceptors, so that's why you won't see anything there. Fovea has a lot of cones. Your choroid has blood vessels that nourish the eye. Your retina has all these photoreceptors. That's what I'm missing. Yeah. Okay. And that's all here in words. So your book says it as well. I just kind of put it all together onto one page because it'd be more convenient. So I'll just kind of give it a second for anyone who wants to look at this, read it, screenshot it, whatever you want to do. And then we'll go on to next. Okay, so photoreceptors. So this might be a little confusing. Just understand what happens when there's light and what happens when there's no light. So let's see. If we start with this photoreceptor, it has 3 parts. It has an outer segment, inner segment, and synaptic terminal. Synaptic terminal, anything would release neurotransmitters. And I put some things there. Okay, so for this light and dark, I have a really good image in the next slide. So let me get to that, but let me kind of read this first. So in the dark, your retinal is in this inactive form, and it's called cis. Now it's going to release neurotransmitters, and that'll cause depolarization, and it'll suppress your bipolar cells. Light is the opposite. So I'll just kind of show an image on the next slide that I think is much better. I'll let whoever is doing whatever continue. And then she has this mechanism for the light in terms of like the G protein. I don't know if you need to know that in detail. I kind of think it's too much for the level that you guys are at because I'm currently learning that in my molecular cell biology class. So I can kind of make it super simple. Okay, I love this image. I think it's great. So if we start with this one here in the dark, this is going to be cis. And then your sodium channels are always going to be open in the dark. So if it's always open, that means sodium is entering your cell. Sodium is a positive ion. It's going to make your cell more positive. So if you understand those action potential graphs, this is kind of what it's showing here. It's becoming more depolarized. And then it's going to be releasing glutamate. There's a reason why it's always open and always depolarized, and then I will get to that later. But kind of understand the difference between light and dark. For light, it's going to switch to trans. It's going to turn on this whole GPCR system, which will be right here, and I'll try to cover it super simple. But it's going to close those channels so that no more sodium is going to enter. That's going to make your cell more hyperpolarized because no more positive things are entering your cell. So understand that. Okay, so for this part, this is very much a signaling cascade. So if you remember back to your endocrine system, hydrophobic versus hydrophilic, the way it transmits a signal, this is similar to hydrophilic. So basically, a light is gonna hit. It's gonna switch from cis to trans, as you guys know. It's gonna activate this G protein, GPCR thing. So, okay, wait. Right here is what is normally in the dark. Okay, so your sodium channels that are open when in the dark, it is able to be open because of this thing called cGMP. So you need cGMP for it to open. So the whole point is, if you have light hitting, it's going to take away the cGMP and turn it into something else. So it's not even going to be cGMP anymore. So by decreasing the amount of cGMP, your channel is going to close. So that's why no more sodium can enter, because this whole system of light is basically going to decrease the amount of cGMP that binds to close your channels, and you're going to hyperpolarize. I hope that made sense. I try to make that super simple. That's basically what the whole idea is happening here. Your phosphodiesterase, this protein, this enzyme is going to decrease the amount of cGMP so that you have less, and then it's going to close those channels because your channels need cGMP. Okay, gonna move on now. Okay. So from your photoreceptors, it's going to synapse with all these different cells in order to get to your auditory cortex or nerve, or optic nerve, really. So you have something called bipolar cells, ganglion cells, horizontal cells, and amacrine cells. So these are just kind of descriptions of where they are. So I'll kind of go more in depth with that in the next slide. So I'll kind of let whoever is taking these notes to finish whenever they want. Okay, I'll assume they're done. Okay, so this is these are both the same thing. I like this image better just because it's really simple. This one looks very crowded. So basically, you have your photoreceptors. That's your like rods and cones. It's going to get that signal and transmit it down. So in a direct connection to the optic nerve, which is where you want it, is really going to be photoreceptors, bipolar, ganglion, optic nerve. That's kind of like the main direction. But these other ones kind of help regulate the in-betweens. So the in-betweens, so for photoreceptors, you would have your horizontal cells that kind of regulates and mediates between them. And then you have your amacrine cells that kind of regulate and mediate between your bipolar cells. Those are the in-betweens, but in terms of direct connection, it would be photoreceptor, bipolar, ganglion. I kind of did that here. So you have light hitting your photoreceptor, bipolar, ganglion, optic nerve. It's going to go to your thalamus, which again, that's where your sensory relay information is. It's called you don't need to know that. Anyway, so it's going to go to your visual cortex. In terms of the other situation that we talked about, when light hits, it's going to go from cis to trans, activate all these other things. Ultimately, you're going to close your channels, hyperpolarize, optic nerve. And then no light, same thing. So the whole idea of it, the channels always being open, this is what Dr. Wilson said. So it's going to prevent misfire and increase sensitivity till when light does occur. So that's the reason why your sodium channels are always going to be open in the dark. Like there's a reason for everything. And that's kind of what it is there. Okay. I think that's it for vision. So we're going to get into chemoreceptors now. Okay, so smell. Your smell is a little different when it does not relate to the thalamus. So this one's a little different. I think this might be the only one that does not relate to the thalamus, which is weird, right? I personally don't know why. That's kind of a lot of detail, so I'm not going to even try to go there. Okay, also your smell has the most amount of receptor proteins, because if you think about it, you have so many odorant molecules out there in your surrounding. So you have to be able to have different receptors to be able to recognize all of those. So that's why it kind of has the most. In terms of this image, so if we look here, you have your odorant. It's going to bind to its receptor. It's going to go through this whole G protein coupled receptor that we saw earlier. Basically, that means it's involved with a G protein. So that's going to happen once your G protein does its stuff. It's going to create depolarization. That's going to send that signal, and then it's just going to pass through all this to get to your brain. So it's going to get through here, pass through the holes in the bone, get to the olfactory bulb, synapse there, and it's going to make its way to where it's supposed to. I don't know if I put where. Oh, okay, yeah, you have an olfactory cortex. Yeah, there's a spot for all these things. It is a part of the limbic system. That's a really good thing to know. So kind of know about that also, because that's involved with your nervous system chapter, or your nervous system. Yeah. And that's because, so for example, say you smell something, you might get triggered and kind of remember an experience or some emotional moment that you had. So that's kind of why it's tied to your limbic system. I did all this here, so I'm just not going to say that again.
Are the hairs, like the one, like whenever they bend, does that trigger the depolarization?
For this case, yeah, it says hairs of olfactory receptors here. I think it's a good assumption to have that they'll have some sort of hairs because there has to be some reason for it to like transmit through.
And the olfactory cortex is in the parietal lobe?
Probably. Parietal lobe has all the little sensory things. So yeah, I would assume so. Because
They
I know
said-
what the lobes do, right?
I think temporal is hearing, so parietal is probably, yeah.
Yeah, parietal is kind of your sensory awareness and all these different things. Yeah, that's kind of in the beginning of my slide. So if we have time, we'll probably go back to that. But definitely know what each of the lobes do. They're very unique in each of their functions. Would olfactory bulbs be involved with the hippocampus as well? They're both in the limbic system. They're both separate structures. I'm sure they're connected somehow because mostly everything is. So if you think about it, say you do remember something, you may recall a memory from your hippocampus. So your hippocampus is dealing with memory like short term and long term. So maybe if you do get that, some other signal will cause you to remember some kind of memory. And yeah, I would say yes. In short terms, yes. Okay. Okay, so now we're going to talk about taste. I think this might be the last two sentences. You're welcome. Okay, so the way I did this was in terms of levels of organization. So I like to start broad. So I started with your tongue. From there, you have little papillae on your tongue. Inside your papillae, you'll have taste cells with taste buds. And then as you can see here, again, there's an afferent nerve. All of these things connect through this nerve to go to your brain to be integrated and processed. Okay, I did that there. You have 5 taste receptors. Sweet can detect sugary things, sour, salty, bitter. Umami is might be one thing that you are unfamiliar with, but that one detects glutamate, and that's an amino acid. And for example, that's in your MSG. It's monosodium glutamate. I learned this in biochem too, but I can't remember. And then these are the different ways that they kind of will perceive that message or different receptors for it. So as you can see here, the sweet, umami, and bitter, they go through a GPCR system. Because again, there's this GTP and there's this G protein. A GPCR means it has these spanning integral membranes. So that's kind of one way to remember that recognizes GPCR. And then these are just normal channels. If you guys, this might be something that you may see. So what kind of channels are these? So there are 2 types of receptors. It's like blank tropic, and then the other one's also blank tropic. Which one does this look like? I think I talked about this beforehand, but this would be a good way to also learn and recognize what that means.
Ionotropic?
Yeah, it's ionotropic. Ionotropic means it will bind directly to it, whereas metabotropic, it's going to activate something else. Like a G protein is more metabotropic. I have that slide somewhere else, but that's kind of another way to recognize and incorporate other things that you're learning. Ionotropic is more direct. It requires only one, whereas this is going to activate this, but this G protein is going to activate another protein. So that's why it's kind of like more steps, and that's metabotropic, metabotropic. I don't know how to say that. But yeah, so this is for your taste. Oh, cool. Okay. Now we're going to go on to thermoceptors and nociceptors. Okay. All earlier was mechanosensors then. Okay, I didn't put that together, but that makes sense because of hair cells. Okay, so you have thermoreceptors. These are detecting change in temperature. Free nerve endings. So if you remember back when I talked about those mechanosensors, free nerve endings are the ones that are at the surface. Those can detect pain and temperature, and you can kind of see this here as well. Snakes have something called pit organs. That might be some random thing she may say. Hypothalamus, know what that does. That's part of your central nervous system. I'm trying to read this, remember. You warm receptors, cold receptors, it's kind of like a duh. You're going to have something that detects hot, you're going to have something that detects cold. This is just an image. Yeah, okay, that's not really much to say about thermoreceptors. I feel like that's kind of, it makes sense in terms of like if you're cold and you're hot, your hypothalamus will work, and then, but that's all detected by your thermoreceptors. But yeah. Nociceptors are more of tissue damage. So let's see, let me read this. Oh, these are involved with your mechanoreceptors, thermo, and chemoreceptors, and those are kind of like all tied together to help realize that pain's going on. Because say, for example, you might eat something and it might do something to you. I don't know. There's like a lot of ways that you can integrate all these together. Examples would be violent forces, of very hot and cold temperatures, because those can be painful. Dangerous chemicals. So chemicals will be tied with chemoreceptors, temperatures, thermoreceptors, forces somehow, some kind of pressure will act on your mechanoreceptors. That's a good way to tie them together. Yeah, good note for glutamate and umami. Make sure you do know what these neurotransmitters are because she said to not memorize them, but then she was like, oh no, this part and this part and this part of them, which basically sounds like memorizing to me. So find some way to remember that. This might, if you like to associate things, glutamate and substance P, there's more for that you can use this and tie it together to remember. You have something called endorphins. It suppresses your pain. I'm sure you guys have heard of that, like a runner's high. After you run, you get these endorphins released and you feel better. I think endorphins is one of your things as well. Okay, that was it. Okay, so for magnetoreceptors and electroreceptors, I think this is the only slide I have for it. There's really not much to say. Magnetoreceptors are really helping animals migrate to different locations. I put some examples there. They can detect the Earth's magnetic field. It's different in different regions. Yeah. Electroreceptors, they detect electrical fields. So like sharks and bony fish can do that. They can locate prey, navigate around.
Is this the one where they like stun their prey? Like if it's like active and passive?
I think for electroreceptors, yes.
Okay.
So, yeah. But again, read up on that if you're like not sure. I'm just kind of putting these topics out here so you guys know and recognize them. Okay. Okay, so I think we're getting to the muscles now, and this is the most recent unit that you guys have done. Let me see. Okay, yeah, I don't think I have too much for muscles. Okay, so normally I use this as a pop quiz in my sessions. I like to like blank things out, and then if you can kind of recall what they are, that's a good way to test yourself if you would like. Know the difference between ligaments and tendons. So ligament is bone to bone, tendon is bone to muscle. Find some way to remember it, unless it's easy for you. How I thought of it, I know this guy who has the last name Ligon, and then he was very bony and athletic, and he runs a lot. So then I just assume bone to bone because it's all bone. That's how I remember it. Different ways to remember. All these different bands, I'll find that I have a slide that explains a way to easily remember these different letters. So tune in for that, which will be soon. And then this is basically showing how you get a nerve. So this will be your efferent, like I said. So it's exiting, so it's going out to your muscles to tell your muscles to do something. So this is just going to show a way going from your neuron to your muscles. So we'll get into that later. I kind of just did the difference. This is more back to your tissue chapter. Focus more on skeletal, really. I just put these differences if you guys would want. But this is more for skeletal. Know what skeletal is and its different things. So you know its functions for locomotion, moving body parts, and maintenance of posture. But you can kind of see how this is different from these two. This is voluntary and these are involuntary. Your smooth does deal mostly with blood vessels and intestinal linings and stuff like that. Cardiac, think muscles in your heart. I'll give this a minute. Okay. This, I just got an image from your book, but this is also a good way to know what they look like, description, location, function. This is in that chapter back in tissues. I can't remember what number it is. So yeah, that's where I got that. Okay, so I did the same thing here. It's levels of organization. So I start kind of broad. So I start with muscles. Within muscles, you have something called fascicles or fasciculus. I think that's plural for fascicle. Within your fascicle, you have muscle fibers. Within your muscle fiber, you have myofibril, and then your myofibril is made up of actin and myosin. These things in parentheses, sarcolemma, sarcoplasmic reticulum, and sarcomere. I just didn't want you guys to get confused in all these different sarcos that you hear. So this is kind of where they are in terms of this. I'll have an image to make more sense of it. Sarcolemma is basically the plasma membrane, but for your muscles. Sarcoplasmic reticulum is the thing that stores calcium. That's similar. It's made up from your smooth endoplasmic reticulum. So again, the whole reticulum, the whole function would be like to store, release, and things like that. That's kind of a good way to remember that in terms of the word reticulum. Sarcomere is, if you remember back to all those bands and lines, your sarcomere is like the unit of a muscle, really. It's defined by the Z bands. So the Z bands define the boundaries of what a sarcomere is. Yeah, T-tubule also. I'll get into that, but definitely know what that is also. Okay, I think, so this one's more specific for muscle fibers that I put here. They're just held by connective tissues, merged with tendons. They get all that, and then they're packed with myofibrils, as you can see here. So within a muscle fiber, you have myofibrils. I don't know why I organized this so weird, but yeah. This is blurry. Okay, so I'll kind of get into the way that I remember. If anyone else has a way that they remember, you are free, and you're free to share it with anyone else. So, how I thought of it back when I took this class 2 years ago, which is kind of a long time. I'm a senior, so I like to start out broad. So I kind of go out to an edge. So I choose this as an edge because this defines the sarcomere. So I start out here, and then I try to go in towards the middle. So this is your Z line. The next thing that I see is your I. The next thing looks in this section is A. Then you get H and then M. So you can make an acronym to try to remember that. So the one that I looked some up because I didn't use acronyms exactly. I just remembered Z, I, A, H, and M. But there's one called, let me type it. So here is a hungry man. So it's all this there. So if anyone thinks that's cool, then you can use that. Z here, they just use it to see the wording, so it kind of may trigger it. But again, it's Z, I, A, H, M. That's one way to remember it. If you think a different way, I thought of this one last minute. So if you look at I, the shape of the eye is super thin. So you can think of thin filaments. It only has thin filaments. For A band, it kind of looks like a tent. So it's kind of covering everything. So it has thin and thick filaments. So the way the A shape looks like a tent, it's going to cover everything. H band, it's thicker than I, it's fatter. So you can say thick filaments. And then M line, I feel like that should be kind of more obvious. It's just going to be the one that's it's aligned directly in the middle. Z line, that kind of defines a sarcomere. So let's see what else. Yeah. So if anyone else has a way that they remember, you're free to share it with everyone else. But this is some couple ways that I found and some that I do myself. And then I just kind of put it into words here. The same thing. Okay, next. Same thing. Oh, I mean, I guess we can get into here. Hold on. I had to lock my door. People were coming home. Okay, so here it's the same thing, just a little more broad out. So if you look down here, just kind of recognize thin and thick filaments. And then the one that has the troponin, the tropomyosin, is the actin. Let me see if I have slides on that one. Okay, yeah. So let me go to the next slide. Okay, so you have your filament. So this should be thin filament. I think I messed this up when I made this kind of fast. So if we look here, okay, so for your thin filament, like I said earlier, is your actin, and it has troponin and tropomyosin. So tropomyosin is going to be this long green thing. It has the word myosin. It's going to be covering your myosin binding sites. That's how I think of it. Troponin, they're the ones that are just like more discrete locations. So that's kind of where your calcium will bind. So your calcium will bind to troponin. Troponin will allow tropomyosin to move out of the way, and then your myosin will bind to where it's supposed to bind to cause a contraction. This is kind of for cross bridges. So this only happens with calcium present, right? Which will bind to the troponin. So if you have a lot of calcium in surrounding, then it's more likely of a chance for it to bind, and then things will happen. If you don't have a lot of calcium, first of all, how is it going to be able to bind? There's not going to be as many binding chances for it because there's not enough around. The chances of it actually binding to where it's supposed to will also make things more difficult. So that's why if you have less calcium, your cross bridge you won't make as easily as if you have a lot, and that kind of makes sense. So I just kind of wanted to put it out there just for it to, yeah. Caffeine I don't know about increasing calcium. It probably does downstream somewhere. I know that caffeine's like is a diuretic. So that's a good fun fact to know. Now I know more. Let's see. Okay, so now we're going to get into muscle contractions. I'm not going to read this because you guys obviously can read as well. So I'll just kind of go into my notes on this. I made a dumbed down version. This is basically in words what it put there. So okay, this is my dumbed down version. So I just found this super simple picture, but what I do, I like to make things a little simple. Once you understand the simple things, you can kind of add more to it to make more sense. So when I think of ATP, when you have ATP, that's going to allow your myosin to let go of actin. So ATP is let go. When you have ADP bound, it's going to be attached to the actin. When ADP is released, it's going to create a power stroke to move it, but your myosin's still going to be intact. And then same thing. So this is kind of like the simple version, and then you can kind of make it more broad. So if we bring it back to, yeah, I made this. It's kind of very dumbed down. I'll wait till whoever's done with that, and then we'll go backwards a little bit. Oops, sorry. Okay, so if we go here, we can even look at this picture. So it's so if we start here, your myosin is bound to this actin. When you have ATP, it's going to let go. So like I said, ATP, let go. Once you have ADP bound, once it gets hydrolyzed is adding water to break bonds. So your ATP is going to turn into ADP. So once that happens, you're going to have ADP. And then when you just have ADP bound, that means you've made this cross bridge essentially. So ADP cross bridge. Once your ADP is let go, then you have a power stroke. So no ADP, power stroke. That's kind of how I think about it. And if you kind of keep that in mind, you can put it together and create this muscle contraction. The next, we'll talk about it going from a nerve to your muscles. So I'll kind of let whoever do that finish. Okay, next. Okay. This is in your book, I'm pretty sure. So it was kind of big, so I had to do it into pieces, so it kind of looks weird like this. But so again, so you get an afferent signal from some stimuli outside. Okay, so it's going to go to your brain, integrate, it's going to send it out to your muscles. So this is the out message, which is your efferent nerve. So your signal is going to go down to this nerve. It's going to release these acetylcholine and neurotransmitters. And then this electrical impulse will occur, and this is your sarcolemma. So it's going to go through this sarcolemma, and then it's going to get inside to your sarcoplasmic reticulum. So that's the whole system or level from sarcolemma, reticulum, sarcomere. So it's going to go into this thing called a T-tubule. So a T-tubule is kind of connected with your sarcoplasmic reticulum. It's just kind of a way for the electrical impulse to travel. So the thing with a T-tubule, it stands for transverse. So it's going to kind of dip in, and the dip in is going to allow for a deeper transmission of the electrical signal. So it just allows it to go deeper into your muscles more, so it can kind of spread more. The signal will travel through your T-tubule. With that, you will have these calciums be released from your sarcoplasmic reticulum. That calcium, again, being released, will bind to troponin. The tropomyosin will move, myosin will bind, and everything else. So that's kind of how it is if you think about it from your central nervous system out back to your peripheral nervous system to your efferent nerves, and that going down to your muscles. And then this is another image of it as well. Oh, for acetylcholine, that's one of the neurotransmitters that you need to know as well on that chart. So you can also think about it. Acetylcholine helps in movement, and then it deals with the whole neuromuscular junction for its location. So your neuromuscular junction is right here. It's just the space between your nerve and your muscle, hence neuromuscular junction. Yeah. I basically said the same thing. This is just kind of it in words, so I don't want to read that, but you can take pictures. This is talking about your sarcoplasmic reticulum. We talked about this. So again, your plasma membrane, that's your sarcolemma. Then if you go in deeper, you have your sarcoplasmic reticulum. And inside your sarcoplasmic reticulum, these different bands, your sarcomere, which is defined by each of those Z lines. This is the same thing, level of organization. I said all this earlier. So yeah, what's next? Okay. Okay, so you have these drugs. You have some two main ones that kind of deal with acetylcholine release. So you have one called botulinum toxin, and then you have Curare. They both have the same outcome, but their mechanism is different. So for outcome, you will end up being paralyzed, and you may die from respiration failure, and that's because of your diaphragm. That makes sense because your diaphragm is a muscle. These drugs inhibit muscle movement. Your diaphragm won't move, thus you won't breathe, and then respiration failure. Okay, so for botulinum toxin, it blocks the release of acetylcholine. So inside your nerve, those vesicles have these neurotransmitters, aka it would contain like acetylcholine. It just won't be able to be released. So that's with botulinum toxin. Curare, the acetylcholine will be released, but it won't be bound to the receptor. So it's probably some kind of thing that will. So Curare may bind to the receptor itself to not allow acetylcholine to bind. That's kind of one way for it to do that. And they both have the same outcome. You also have rigor mortis. That means you're not making any more ATP, so your cross bridge is locked. Because again, you need ATP to let go. So in this case, myosin will not be able to let go, and you'll just be stiff. Yeah. Yep, that's it. Okay, what's next? Okay, we're almost done with this, so I may or may not have time to get into the other stuff. So this is single twitch, twitch summation, and tetanus. Single twitch, so you'll go up, but then you'll have relaxed fully already, and then you'll do it again. It's not going to be as strong as twitch summation or tetanus because you've already relaxed down to your ground state by then. You need to be able to have another signal hit before it relaxes ground state. You can increase the strength of the signal. Tetanus has the most. Okay, fast twitch and slow twitch. We'll get into that. Know what myoglobin is? Basically, it's just kind of like this blood system, oxygen storage for muscles. Myo is muscles. And we'll get into fast twitch and slow twitch. Yeah. Same thing here. I said this here as well. Yeah, muscle twitch is a weaker one. Okay, next. Okay, so now we're just going to talk about each of these fibers. So slow muscle fibers, they contract relatively slowly. They have a low intensity. Okay, you guys can read. I'll just kind of say things that I feel are distinctive. So each of these have their own kind of thing. So the things I underline, I feel like are certain traits that are specific that she could kind of confuse you with. So just recognize which one has a lot of mitochondria, which ones have more oxygen or require more oxygen. So these have more mitochondria just because they require oxygen. So if you remember back to cellular respiration, you need oxygen to be able to make a whole bunch of ATP. And that last step of the electron transport chain, which is in the mitochondria. So that's why they have more mitochondria, they need more oxygen. And slow muscle fibers utilize oxygen as well. So like I said here, they make ATP. That's with cellular respiration. They have a lot of myoglobin. The myoglobin makes the color the way it is. So that's why it's so red. It's because it has more myoglobin. And this helps with things such as endurance, marathons, anything long distance, and that requires endurance because you can do it for a longer periods of time. And that's kind of the whole point of your slow muscle fibers. It helps with sustaining your energy. Yeah, know all those traits. Fast aerobic muscles. So aerobic means that they need oxygen, but in terms of it being fast, its contraction is quicker, it's more intense. They have mitochondria as well because, again, oxygen is being used. They are pretty red also because of myoglobin. ATP because they do use oxygen, like I said. Ooh. Sorry, I just ate. Let's see. Yeah, fatigue. So in terms of putting in these each into like levels, so it would be slow, fast aerobic, and fast anaerobic. So fast aerobic is pretty intermediate in terms of all these different things. So like I said here, fatigue, that would be in the middle. Capacity for making ATP in the middle. So this one's the one that's kind of in the middle. And then lifting weights was an example, she said. Fast anaerobic, they don't require oxygen. They have few mitochondria because, again, few oxygen. They don't have a lot of myoglobin, so the color is more pale, just definitely not as red. Limited supply of blood. Okay, so they make ATP by anaerobic respiration, which is glycolysis. I don't want to give you guys like a Bio 1 review, but for those who want it to be more clear, you have glycolysis. So your cellular respiration comes in 3 steps. The first one is glycolysis. Glycolysis is the one that is the one that does not require oxygen. You can still make ATP, but you don't make a lot as compared to if you get all the way to the electron transport chain, which you'll make like 3 to ATP or something like that. But that requires oxygen. It cannot use oxygen, so that's why it's more bound to glycolysis. And then you can get cramps and stuff like that because you have all this lactic acid. Anyway, I'm a bio major, so I can go on forever. This one's more susceptible to fatigue. Yeah. So again, be able to put these in like different levels of like what they would be. Yeah. And all these fast muscles help short term, especially for animals to escape prey. That's a good one. Forgot about that. Okay. Yep. I just put it here. So what I put earlier in those individual slides were from Dr. Wilson's slides. And then I put this together, and this is from your book. So relatively same information, maybe 1 or 2 things different, but again, it's the same thing. Yeah, the book mentioned more of like the muscles in birds able to escape quicker. That's the same thing as everything else I said. This is kind of a chart. This is a good way to kind of test yourself if you know each of the differences. Like I said earlier, fast aerobic, a lot of it's more intermediate if you get confused. But also keep in mind the oxygen that can tell you about the color, mitochondria, ATP-wise. So this is a good chart to use. And then this kind of also just shows your slow twitch versus fast twitch. Like I said, distant runners who can go, like for their endurance, go longer distances, they have more slow twitch fibers. You can see by all the red. So color is a good indication. Know the color. Sprinters, since they only can do like a certain distance for a shorter amount of time, they utilize their fast twitch muscles more. This was just a review of Bio 1. So, yeah. Okay. So I said this earlier, motor units are more efferent because again, it's going towards your muscles. All muscle fibers in a motor unit are of the same type, but and contract together. You guys can read. Your cerebellum is very much associated. Your cerebellum, know what each of your brain structures are associated with. Cerebellum helps with like coordinating your motor movements. So that's pretty important in terms of knowing and telling what your muscles what to do. Okay, what's next? Yeah, so that's more back to nerves. I think I might cover that. So wait till I get to the end. Yeah, I think I have that somewhere. So I'll keep note of that. Let me get through muscles. This is just kind of another thing as well. So your muscles are able to coordinate which, or if anyone else can answer that question also, you can do that. I'm going to try to finish my slides real quick. So for muscles, it's able to regulate how many neurons or what neurons are going to like cause this potential. So for example, if you need smaller amounts of movements or like weaker movements, it'll like coordinate like these two. Whereas if you need stronger contractions or movements, it'll coordinate more. So this idea is just that you have multiple neurons binding with different things. Okay, so now we're going to start talking about the different skeleton systems. Okay, yeah. So this is for your hydrostatic skeleton. Okay, so I'm going to explain how this kind of works before I move forward. It has these muscles called circular and longitudinal. So I'm going to try to describe this as well as I can. So your circular goes like this, but when it contracts, it's going to make it kind of look like this. It's going to kind of squish the circle inward, whereas longitudinal is it's going to go like this. If that makes sense. So, okay, so if we start with first your the circular muscles will go like this. Once the circular muscles contract, it's going to make the length increase. So as you can kind of see here, it's circle, then you go like this, it kind of increases in length. Then it's going to hook down onto the ground or hook down somewhere. So this part right here, it's going to hook to the ground. Then your longitudinal muscles will contract and allow it to pull itself up, and that's going to shorten the length. So that's kind of how it's going to move, if that makes sense. So it's going to like squish to get longer, hook, increase, and that's how it's going to move. I hope that was like a way to describe it, but that's kind of how your longitudinal and how your circular muscles kind of work. So it's more a circular hook and then longitudinal. And this is just kind of what it looks like. And I have all this in words, so don't worry. Yeah, so hydrostatic, hydro is water. So basically in its body, it just has all this water. Static, it's not really, the water's not going anywhere. So it just has all this pressure built up inside of this organism. Yeah, I mentioned those two. There's some examples. Yeah, it's not just in bugs, it's also in humans as well. Yeah. Okay. So we're going to talk about exoskeleton. So exoskeleton is basically when they have, I guess you can say their skeleton is on the outside. So exo. Sorry. So yeah, so it's like this rigid structure on its outside, whereas compared to endoskeleton, like us, for example, we have our skeletons on the inside covering our internal organs. For exoskeletons, they have this outer one covering their whole body, really. And anything that has like this hard calcium, like rigid substance, that's kind of them falling under exoskeleton. Yeah, so there's a lot of benefits for them as compared to hydrostatic organisms. So these are some, I have it on the next slide, but I can cross-check you on that. Or I can wait and I'll just show it on the next slide. Okay, I'm gonna have to move on, we're cutting it close. So these are some things for exoskeleton. Basically, this one's better for them because less fluid, and less fluid means they'll weigh less. So it's easier for them to move faster. They can retain moisture, and it'll protect them from dehydration, as someone said earlier. It provides some armor for predation, and these are some different examples. Next, endoskeleton, not much to say, but I'm going to go more in depth with it. Basically, the skeleton's inside to protect the internal structures. Let me see. So these are kind of the functions of this. Just supports calcium storage, blood cell formation. That's kind of with your bones, really. Has all that. Yeah, humans have 206 bones. You have an axial and an appendicular section. So when I think of axial, it's more of just anything that's kind of down the middle. So what is it? Your sternum and your ribcage, it's all kind of like down the middle, and your cranium. Your appendicular is more things that are kind of like sticking out more. So it's a cute picture. Your shoulders kind of stick out, and then your pelvic girdles and your limbs, your arms and legs kind of stick out. So those would be considered your appendicular. Okay. Next. Sorry about the picture. We only have so much time. Okay. Your bones. So you have compact bones and you have spongy bones. I think I have a picture on the next image, but this kind of ties with your endocrine system as well. You have osteocytes, osteoblasts, and osteoclasts. Osteocytes are just mature bone cells. Osteoblast builds bones. So if you want to think about it, in order to build a new house, you can kind of demolish your old house and build a new one. So that's kind of bone building. Osteoclast breaks bones. So find some way to remember that, but you can. Ossification is just kind of building more bones. I'll show a picture of compact and spongy in the next one. So your spongy bones kind of on the ends of bones also, but they're also in different locations. I can't remember at the moment. But the outside here is also compact bone, and then your spongy bone has all these like things. You can also have bone marrow here as well. Okay, and this yellow is your yellow marrow. Anything that's yellow, really, you should think of fat or adipose tissue. That's kind of what the yellow signifies. So your tissues, let me see, red marrow, you can get red blood cells from it. That's also known as hematopoiesis, making new red blood cells. You can find them in a lot of your flat bones, but as well as your long bones, if we're looking at this image here. Yellow marrow is for white blood cells. Yeah. Okay, that's it. So different joints. So she didn't really talk about fibrous, but if you want to incorporate that, so you have synovial and cartilaginous that she talked about. This is the most movable. This is semi-movable, and this is immovable. So that's kind of how the ranking of order is. So we're going to start with synovial joints. These are things that kind of have more freedom for movement. You have a hinge joint and a ball and socket joint. They move differently. So hinge joints are like your fingers that kind of move one direction, whereas like or maybe not fingers. some example for hinge joint where it moves like in one direction, like your elbows kind of just go this way. Your ball and socket joint, think of like a ball, you can kind of twist it around. So you can go like this, this would be a good way, like any way you have free movement in any direction is a ball and socket joint. Like your shoulder, I can move it around and around. What else? Okay. That's all I really want to say about that. But I put it all here, so. And this is kind of another image of that. So you have those ligaments that was mentioned in the slide before. It kind of holds all these things together. This is a ball and socket joint, and this is a hinge joint. So this one has more freedom of movement in terms of like 360, just because of that. Yeah. Cartilaginous is in your spinal cord. It's in between each of your vertebrae. It's less movable, but more movable than fibrous joint. They are also covered in cartilage, but they don't have a fluid-filled capsule, whereas synovial does have a fluid-filled capsule. Yeah. Fibrous, I'm not going to get too much in this. I don't think she mentioned it, but like on your brain, in your skull, you have like these weird irregular shapes. That's kind of one way if you want to see fibrous, but again, it's immovable. We obviously cannot move the bones of our skull. If you can, let me know. That'd be kind of cool. I don't know what's going on there. I have to move on. I'm sorry. I think this might be the last one for muscles, and then I might start up at the beginning, but only for like 5 minutes. Your muscle-bone interaction, agonist is just allowing it to move, really. Antagonistic pairs is a little, it's just kind of going in pairs. So if you think of your biceps, bicep, tricep, I don't go to the gym, but I know that much. So extensor, for example, if I go like this, I'm flexing my biceps, but my tricep back here is extending out. So extending would be like it's stretching more, and then flexor is it contracting. So that's an example of antagonistic pairs. You have something called a fulcrum. That's kind of at each of the pivot points. So like a fulcrum here and here. It's at each of the ends. And I think that's it. Let me see. This went by so fast. Oh my gosh. Okay, I'm just going to go through the other stuff really fast, so I may not even read it all. So I'll just kind of stay for like 5 more minutes. And let me start from the beginning. Oh gosh, so many slides. Almost there, I promise. Okay, finally. Okay, so this was kind of the pathway for you. So now we're at neurons. So this is like the first unit that you guys learned. So the basic 3 components of it that was in your book at least is reception, integration, and response. So I kind of just put that there. You're welcome. So this is your afferent and efferent that we talked about earlier. Okay. This is just parts of your nerve. Know that each of the parts. Interneuron is something that's closer, and projection neurons go out a little further. These are all your glial cells. Only know these first 3. These other 2 I just found in a book for my study. So just know what these 3 do. Schwann cells, and this should be oligodendrocytes. They have the same function, but just different locations. Know what your myelin sheath is for, and then your nodes of Ranvier. Understand this. This for sure you should understand. Know what each part is, know what's going on. So this one I talked about in terms of the gates. So what gates are opening, what gates are closing. These are just some other notes that I made of. And then know like depolarization, repolarization. Your threshold is important. If you don't reach this threshold, you don't create an action potential. If you do pass it at any way, if you pass it here or if you pass it here, either way you get an all or nothing response and you will get an action potential. So it's an all or nothing. If you don't reach this threshold, you don't make it. You need to reach this threshold. It's important. No negative 70, that's your resting membrane potential. Your refractory period starts from here to here, and that just makes sure that you don't go in reverse, but I have this all here. So these are the steps of your signal transmission, stimuli, threshold, depolarization, repolarization, and all that. This is just an image showing the gates. If you need more understanding, it's on your book. I think I said the same thing here. The magnitude is always the same. It's an all or nothing response. you have your EPSP and your IPSP. I have this slide, this is better. Yeah, your EPSP are depolarizations that occur from activation of synapses, and IPSP is hyperpolarization that results from activation of synapses, but that hyperpolarize. This is more of it shows summation, temporal, spatial. Temporals from the same EPSP, spatials from different EPSPs. EPSP and IPSPs can cancel each other out. This shows that it kind of happens as it goes along. It doesn't skip from here and here. It kind of has to happen in this manner that goes down linearly. Salutary effect, know what that is. These are differences between electrical synapse and chemical synapse. This is a direct connection where they actually touch, and these are just diffusing through a cleft. This is the difference between ionotropic and metabotropic. Know this. Here's a little YouTube channel if anyone's interested in the link. It kind of helped me understand the differences better, but this is more like the ligand will bind with this receptor, done. The other one is metabotropic, and ligand will bind this receptor, but this receptor will have things happen to act on a different protein. For example, GPCR is an example of metabotropic. It requires more than one protein. Ionotropic kind of deals with one protein. It binds and allows that channel to happen. So an example of ionotropic would be ion channels, and that would be a really good example. I know, I'm so sorry. I can't even send out my slides because they don't want us to. So what I normally try to do Wednesdays, I do like half of everything, and then Fridays will be like the other half or before your exam. So I'll send a better announcement for that next time. But again, your TAs have sessions over the weekend. I don't really know how they do it, but you're welcome, Stephanie, you too. These are all your neurotransmitters, just location, action, examples. There's a table in your book, so just look at that. Just look at that, and it's all in there. So I'm not going to go into that just because it's in your book. These are different neural circuit motifs. You have a feed forward, feedback, excitation, and inhibition. These aren't in order, so just kind of be able to recognize. So this one is feed forward, this one's feedback excitation, and this one's feedback inhibition. So you can kind of see with inhibition, it kind of inhibits the one before. This one will continue to excite, and feed forward is literally just this whole, forward system of transmission. Yeah, I put those out of order because normally I like to test people in my sessions. So yeah, I'll give it a minute on that one. These are the differences between all your systems. I think this is in your book, and I feel like you should get a general idea. So here's that. And this is just sympathetic and parasympathetic. This is sympathetic. This is parasympathetic. Just kind of for this, I would kind of imagine yourself being chased by a bear and just think about what your body would do. So for sympathetic, a lot of things will be inhibited because you're scared, but also all your airways or eyes or your pupils will be dilated. So that might be one that might be confusing. So dilation and opening of airways will be sympathetic. Constriction of airways will be parasympathetic. But other than that, everything else is inhibited for sympathetic. These are just talking about parts of your brain. So I'm just dividing it from forebrain, midbrain, and hindbrain. I'm just saying what they are. So just make notes if you don't know these. Not necessarily you have to read this. Just if you don't recognize anything about it, just kind of keep it in mind. This is the same thing. So basically you have a neural tube. It's just this tube when you're an embryo. The front part, the anterior part of that tube will turn into your brain, and that will have more cell proliferation. That's why your brain is so much bigger. The rest will become your spinal cord. And your brain that's forming, the forebrain, will have more proliferation. That's why it's kind of bigger. I don't think you need to know that. I just thought it was cool. I use this to test people. Your blood-brain barrier, it's made up of epithelial cells and tight junctions because, again, it has to create that seal so that you don't have random things going in. That's important. But it has these functions, but basically no toxins, regulates things going in and out. Yeah. Oxygen and CO2, they're like small gases that can diffuse, whereas the bigger things like glucose and ketones require transport proteins. I don't know if you need to know that. But brainstem, vital functions, really. That's how I think of it. I put wakefulness because these three are most elevated when you're awake, and then when you're sleeping, you can see that these are a little more less. I don't think that's important either. This just kind of shows neuromodulation that they kind of go everywhere to modulate different parts of your brain. This is ascending and this is descending. So how I think about it is ascending, you're going from your thalamus to your cerebral cortex. So you're kind of going up, you're ascending, and it's kind of doing things more for your brain. So like sleep-wake cycle, alertness, and consciousness. Descending reticular formation, let me just do this. Descending reticular formation, it's going from your hypothalamus down to your spinal cord. So it's like descending. And as it goes down, I think of it as going to my muscles. So it's going down to my muscles. Anything going up is more for my brain. That's how I think about it, at least. Any way that helps you study would, be great. This is your cerebellum, mostly movement, so I'm just going to keep it there. Thalamus is your sensory relay information. We talked about that. This is where a lot of things go. This is your basal ganglia. So if you don't know what basal ganglia is, make that a note so you can go back and look at that. Because I went over this on Wednesday, so I don't want to go over this too much. Hypothalamus, you guys know what that is from endocrine, just regulates all your homeostatic information. Limbic system are all things kind of relating to emotions is what I think of it. So amygdala is for emotional experience. This might be one way to remember. A hippo hat. So hippocampus and HAT for those. Also your olfactory bulbs. Yeah. Anything that really deals with emotions is a good way to think of it. These are all your lobes. Know what each of your lobes do. Yeah, this is your occipital lobe. This is memory and consciousness. You have two types of memory under long term. So this should be declarative and this should be procedural memory. So declarative, like you can consciously recall, you can declare it. Procedural is like riding a bike. And these are just definitions of consciousness and learning. So I think this might be the last slide. So these are different waves that you have. This is from being awake to sleep. So how I think of it, so beta waves are like when you're busy, that's why they're the most active looking waves. Alpha, you're kind of less active, you're a little more dulled out, so aloof. Beta, I don't have anything. And delta is deep sleep, D for delta. So that's kind of in order from being awake to getting to being sleepy. So I just kind of use these waves. So if you're busy, you'll have beta waves. Aloof, you're a little tired, but not quite. Alpha, whatever for theta. Deep sleep is D. Yeah, so that's kind of one way to remember it. And yeah, I think we ended there. Wow, I literally went through all those slides. So proud of myself. Okay, so that's all for that I have. Your TAs have sessions over the weekend. I think they start at 5, but look at that schedule. If you have any what waves? What is that? Oh, daydreaming, I'm not sure. That'd be a good thing to look up. I know REM, so REM sleep, rapid eye movement, is when you have dreams and nightmares. Daydreaming, I think that might be like theta or aloof because you're still kind of awake, but you're just not as like beta wave alert, So that'd be a good thing to look up. But good luck on your test. Monday, study for it. You're welcome. If you have any questions, you can email me. Again, you have your TAs and they have their sessions. You're welcome. You're welcome, you guys. I think it was a lot of information, so hopefully you got all of that. Okay, cool. Theta waves for daydreaming. I should put that on my thing, but I don't think you guys will need these slides anymore. You too.